So here's our agenda for today. We're starting here at 10 East Coast time. I guess that is 3 p.m. Central Europe time. And we have talks by Cornelia Kuntz, Gary Rosner, and then Christopher Jennison will provide a discussion for that. And then we'll go into two breakout rooms led by Lorenzo Trippa and Richard Elmsley as scheduled here. And the last 15 minutes, we'll have a wrap up and decide what next steps. And this is a Zoom meeting, not a webinar, so everyone will be visible. You can turn off your video if you don't want your pet or children to distract, but that's up to you. You can go to speaker view, and that will mean you won't be seeing the other folks. Everyone should mute their microphone unless you want to talk, and you can use the chat. Ask questions during the talks. Just to say, it might be helpful, since this is really a small workshop, to rename yourself and add your affiliation, like I've done with NIS after my name. And that helps when we're getting into the discussion part. So with that, I will stop sharing and ask Nancy Flournoy, who is the chair of our organizing committee and has done a great job putting these workshops together. So Nancy, welcome from the field of tulips.
Yes, hi. I'm just looking for my slides here. So give me a second. Welcome everybody. Glad to see you. Sorry, I got my time off here on the West Coast, and I had the meeting is happening a little earlier than I expected it to. So I need to know.
If you don't have your slides, Nancy, I can share them for you.
Why don't you, if you can find them faster, why don't you do that?
I think that
looks like the last one. If you scroll to the top.
Okay.
Now,
oh, you're
now I
not
don't see them. Yeah.
The best laid plans. Sorry, Nancy.
Yeah, there. Oops, it's okay.
Okay, that's unfortunately not full screen.
There. Okay.
But, okay.
Okay, so we're very pleased to host this workshop on Bayesian Frequentist Approaches to Rescuing Disrupted Clinical Trials. This is day 6 of this particular Ingram Olkin Forum series. And so before I turn it over to Adam Lane to actually moderate the forum today, I'd like to just set the program in focus for those of you who are new to it, and a little refresher for those of you who've heard me say this before. So the Ingram Olkin Forums are a series that are started to honor Ingram Olkin, who was very strong proponent both of the field of statistics and of the way in which statistics is used in our society. And so these forums are set out to blend these interests of Ingram's to focus on topics where new statistical attention might be needed, topics that are important to the public in general, the nation in general, or the world in the case of disrupted clinical trials at the moment or in general. So the intention is to try to bring new statistical awareness to the issue, in this case, disrupted clinical trials, and to foster new collaborations, new ideas, and stimulate new research in this area, and to come up with action items. So next slide. So we've had several, this is what, the 5th or the 6th in this series. We started out with a couple of introductory sessions from government agencies, statistical leaders in government agencies. And then we moved to talk about bring in people that had expertise in specific areas that we felt were relevant to the topic of unplanned disruptions in clinical trials, with the idea that their expertise might shed light on the state of the art as it's related to this particular problem and where we might go from there, or where the area might go from there. And so we had a session on estimates and missing data. We had one on randomization tests. We had one on coping with information loss and the use of auxiliary sources of data. And following this one, we anticipate having another course on group-sequential designs and sample size re-estimation, and that will really depend on what happens related to those subjects in the working groups that we've got going already. Next slide. So today we are pleased to have Cornelia Kuntz and Gary Rosner give us some motivating discussions on the topic of Bayesian frequentist approaches to rescuing disrupted trials. Chris Jennison is going to then discuss the talks that from Cornelia and Gary. And then, well, before the discussion, we have a short break. And then after the discussion from Chris, we will break out into discussion rooms. And we have, I see I didn't get Richard Hemsley's name in bold there. We have two breakout rooms scheduled, one with Lorenzo Trippa moderating and one with Richard Hemsley. And participants will be randomized to these 2 rooms, with the exception of a couple of note takers and the organizing committee that'll be distributed. The idea, so the main talks are going to be, can you go back just a second? The main talks will be videotaped for the public. The breakout room sessions will not. We will have note takers. The Journal of Biopharmaceutical Statistics is interested in publishing what comes out of the working groups that will be formed after the breakout room discussions. So we're kind of set up for that to proceed. We don't want this session, it's not just a normal session where people give their talks and everybody goes home. We're hoping that you all will stay involved and connected as we go forward. So the breakout room discussions are for you to get to know each other and your interests in this topic, and also then to begin to discuss where the topic might go from there in terms of a research agenda. And there's not a lot of time. We didn't want to have this go on for all day. So we just really expect you to get started. And then we hope you'll join, as I say, I hope you'll join the working groups later to continue working on this topic. So with that, the next session next slide just gives the list of the organizing committee here, and I will be happy to turn it over to Adam to moderate.
Yes, thanks, Jim and Nancy, for introducing the series and a little bit about the session. This session is a bit different than even the ones in the past, because here we have Chris Jennison, who the credit to this session really should go to, has presented each of the speakers with sort of a hypothetical design, and I think I'll leave it to the speakers to sort of discuss the specifics of that, but each of them will sort of give their perspective about maybe Bayesian and frequentist thinking regarding Chris's specific hypothetical design, and then of course I think they'll add details and more generally as well. And then Chris will discuss at the end their talks. So the first speaker today is Cornelia Kuntz from Boehringer. And so Cornelia, if you'd like to go ahead and start, you have a half an hour. Oh, and one last thing. Any questions you have, go ahead and put them in the chat as we move along. Thanks.
Thank you so much, Adam. I hope you can see my slides.
Yes, they look good.
And you can hear me okay, I hope. Perfect. So the title of my talk is Clinical Trials Impacted by the COVID-19 Pandemic, Adaptive Designs to the Rescue, and it's a question mark there on purpose. I'm not saying that we can rescue or want to rescue, but maybe it's possible, and I want to discuss this in my talk. So first, I'd like to say thank you to Chris Jennison, who invited me for the session, and I'd also like to thank the organizers for the invitation and the possibility to present my views on this topic. And I'd also like to thank my co-authors. Last year, we formed a group out of the Adaptive Designs workshop group, basically, and we had very similar questions as proposed by Chris. I will tell you later about what he proposed. So I'd like to say thank you, and I've seen that Sarah at least is here, and I think I saw Nigel briefly at least. So we started setting out a paper, and it's published if you're interested in more of what we have thought about. While I will only give a very short overview of the paper or discuss some specific aspects here. So let me start with the completely fictitious trial design that Chris sent to me and to Gary Rosner. So he wanted us to consider a second-line therapy for type 2 diabetes. The first-line treatment was already unsuccessful. He proposed that there's a two-arm trial, group A and a group B, with metformin and some new drug or some basically standard of care. And he said to us that the sample size was 500 patients per arm that he had calculated. I asked then for some more specific details on the trial. He then sent me the planning assumptions. He has used a significance level one-sided of 2.5% and a desired power of 90%. He assumed an effect size of 0.20 and an assumed variance of 0.95. Randomization is 1-to-1, and then the sample size comes out as roughly 500 patients per arm. And then he went on to explain that that basically 300 patients were recruited per arm by March 2020. This is basically when the pandemic went global, and that no patients had been admitted since that time, but follow-up has continued, and we basically have data for the 300 patients that were recruited so far. So this is the setup, and then he proposed 3 questions and 2 scenarios. So these questions are: would you analyze the data now based on the 300 per arm and draw conclusions? Would you consider restarting recruitment but maybe change the sample size? Or would you change the trial design for an adaptive or a group sequential design? And the 2 different scenarios are that the data is currently blinded, or that the data has actually already been unblinded by the investigator. And now we as statisticians are asked to basically help out on this. And what I liked about it, it's roughly what happened to me in the company. I was basically asked exactly the same question, minus the scenarios. No one had touched the data before they asked me, and I will later come to why this is a good idea, not to touch the data before you ask the statistician. So let me talk about the first option. So the first option is to say, should we just analyze the data that we have at the moment and stop the trial basically early? So here's the frequentist view on whether you can do this or not. So as a frequentist, you would be concerned about type 1 error and about the power. So first of all, the type 1 error is controlled in the setting if you simply stop the trial now based on the blinded data. Statistically, it's basically no different from saying you just had a lot of missing data. So you just didn't collect the remaining 400 patients. And because this is completely independent of the treatment effect or of the data you have observed so far, it's completely missing. and the type 1 error is control. And the power will obviously depend on, well, the sample size that you have observed data for, but also what the true effect size is. And most likely you might expect it to be lower if you assume that the true effect is actually the one you assumed at planning stage. Now, as I said, I wasn't the only one who was asking these questions. I'm a methodology statistician in the company, and I got asked the same questions that Chris sent to me. And then during discussions with other companies, we found out that others also had these questions. And as a result, I programmed an app together with Dongqi from Novartis, which is freely available. It's an AirShiny app. You see the web address down here where you can use the app. And I will use the app basically to explain our thinking, how we would handle such a trial. So first of all, when you start the app, apart from the disclaimer, there is this power evaluation tab, and on the left-hand side, you see the input parameters, which is the one-sided significance level and the desired power you wanted to have at planning stage. And then you have the slider where you can say how much data you have available. So in our case, it was 300 out of 500 per arm. That makes 60% of the data. And independently, we all found out that's basically all you need to calculate the power you will get. The power that I've shown here, the 70% roughly what you get on the available data, is basically assuming that your true effect is the effect that you planned for originally when you set up the trial. Of course, as I said before, the true power will depend on the true effect, but this is unknown. So our best guess for the expected power, I call it, is that you have roughly 70%. And whether this is sufficient or not to stop your trial, whether you believe it's worthwhile the risk or not, will mainly depend on your specific trial. I can't comment on that. I can only tell you with probably 70%, I would personally not stop a trial easily. But given the different options you might have in a pandemic, maybe it's worthwhile trying. So the app also allows you to get an idea of what basically effect you need to reach significance. So when we plan a trial, we power a trial for, in this case, a delta of 0.2, but it doesn't mean that you have to observe an effect of 0.2 to get a significant result. The effect you need to observe in your data is usually much lower. So here you can calculate, originally you needed 60% of your assumed treatment effect. So we have 60% of our delta. So the observed effect in our trial that reaches significance would be 0.12. And now based on the less information on the only 300 patients, we can calculate the new observed effect that reaches significance. And it's slightly higher, obviously, due to the lower data, but it's still below 0.2. So you need an effect of 0.156 to be observed in the data to reach significance. And then you can also look at the relationship between the basically true effect and the resulting power. And you can also do basically the reverse calculation and say, what true effect do I have to assume in order to get back my 90% power? And here you find out that you roughly need a true effect of 0.258. That would give you 90% power based on your 300 patients per arm. And within a company or within our working group as well, we came to the conclusion that we basically, with this specific trial, it depends now whether you think that, for example, at planning stage, you were a bit pessimistic and maybe the 0.2 was a very low estimate of your assumed effect, and you think it could be higher because, as I said before, the true power will still depend on what you assume to be true for the effect. And maybe if you think that you were a bit pessimistic at planning stage, you might stop your trial now and take the risk. Now, this was basically the first option. Should we stop the trial now? And I think it's a bit mixed because there are other options available, and I would always discuss other options first before I would stop the trial. So the next one I discuss here is basically the question whether we should change the trial design. So here, one of the obvious questions is why not change it to a group sequential design or an adaptive design? Here we focus on group sequential designs in the sense of we want to analyze the data now. And if we are significant, we want to stop the trial. And if we are not significant, we want to continue the trial. There's a lot of methodology out there. First question is always for the frequentist would be, how about type 1 error? And basically type 1 error is controlled as long as you pre-specify your changes to the trial before you look into the data. So as long as it's blinded and you haven't looked into the data, or at least not the unblinded data, you can still change the trial design and introduce some interim analysis. And again, the power will depend on what exactly you change the design to. For example, do you use a Pocock design, O'Brien-Fleming, and again on the true effect size. So in the app, you will find another tab, group sequential design. Again, on the left-hand side, we have the input parameters, the significance level, the power, and the data available, or the proportion of data available. And then there's also a choice for different designs. I've only implemented the Pocock and the O'Brien-Fleming design to basically show what happens. Of course, you can change it to other designs. It's just not implemented in the app. And then you see basically a resulting power curve. Just for reference, we put in the fixed sample test. So the blue line gives you the power depending on the proportion of data available if you stop the trial now and just analyze. And then the yellow line gives you the first stage power for the group sequential design, and the red one, the overall power. And here we haven't adjusted the sample size. We still analyze after 500 patients and just wanted to check how this will impact on the power. And as expected, because we have to change our critical values, the power for the first stage is lower than for a fixed design. But overall, you basically nearly reach 90% power. You lose a bit if you would analyze the trial very early. In our case, we basically have 60% of the data available. So our power for the first stage would be roughly 63 or 64%. So it's a bit lower. And the app also will provide a table where you can basically check on the power depending on the data is available. So you see it's a bit lower than a fixed design, but you regain basically nearly all of the power. You end up with 87.5% without adjusting the sample size. You still have 500 patients per arm. Something that wasn't really in Chris's question, but basically was my starting point to look into this, came out of our oncologists. They basically asked the following. So originally, when we planned the trial, there was some treatment schedule and patients were supposed to have a treatment every so often. And let's say they have scheduled 7 treatments over a time period. And they were concerned that when the pandemic started, that basically lots of countries went into lockdown at some point, and due to the lockdown, patients would miss their treatment. And as a result, the treatment effect is smaller than what they originally anticipated when we planned the trial. So here you see that basically a patient would attend their first 3 treatments, but then they missed a few treatments because of the lockdown or some other problems. They had to reach the clinic and so on. But maybe at the end, we solved these problems and the patient was managed to come back, at least for the last treatment. Maybe some patients never come back. Maybe some patients miss only 1 treatment and so on. The main important thing to note here is that we don't believe that the treatment per se works differently because of the virus. We only think that the treatment effect is what they called diluted. So it's watered down due to basically missed treatment, not because something is changing because people caught COVID or something else. And the question was how to handle this and how it will affect our trial. So you see down on the right-hand side, you see that there's an option to say whether you think there's a dilution effect for the second stage. And then you can put in some numbers. 0.1 means basically 10% lower than you expected, or that you wanted it to be at the planning stage. And then what I note on here is basically you see the power curves from before, and the only effect is on the overall power because the first stage data is not affected by the pandemic, it's only the second stage. And then you see that the power will come down a bit, but it's still like only if you have very, very little data, you might make it down to 75%, and otherwise it could still be okay-ish. There's also an option to show the results in a table if you want the specific numbers. So if you look at this here, then we see here the proportion of data available on the left-hand side, and then the power based on the available data. So that is basically for the fixed design. If you stop now, we see the 70% again. And then you see The first two columns then are for a dilution effect eta of 0. And you see that, that we get the 63% and 87 we saw before. And if I now assume 10% dilution effect, I see that the first stage is not changing because it's not affecting the first stage, but I see that I lose another 2% in power. But overall, I've only lost 5% compared to my originally planned power of 90%. And we might find this acceptable and think minor change to this design. I should also say that this is for a POCOG design. We mainly discussed why we would use POCOG here. Usually lots of companies would go for Brian-Fleming, but in this case, in the specific case, we want a very high power for the first stage because we hope that we can basically stop now. And that is basically due to hopefully not running into the trouble of what do we do with the data that's collected during or after the pandemic, because we don't know whether the effects might be lower, we don't know whether the population might be different. So we'd like to stop the data, basically the trial as soon as possible with basically data that was collected before the pandemic. But obviously you can also use the app to calculate for Brian-Fleming, and then you will see that it's a bit lower. So that would be just changing the trial design. And then there was the last question from Chris, should we change the sample size? And here we basically combined that into one, because you could change not just the sample size, but you can change to a group sequential design and then adjust for the sample size as well. And again, if I go back to the app, you can then here find a sample size adjustment tab, where you then have to put in your specific parameters at planning stage. So here you see the original plan was a delta of 0.2, a variance of 0.95, and a randomization of 1:1. And then the sample size is roughly 1,000. And then you can now check what happens to your trial if you want to regain the power. So if you only change to a group sequential design, you don't assume any dilution effect. So you believe that your planning parameters are exactly the same for the second stage. Then you see if you change to a group sequential design, you would need 1,090 patients now to get back your 90% power. So it's not that much. So you might consider doing this. You might also think that losing 2% in power is not really worthwhile adjusting the sample size and just leave it as 1,000. So the next one is you can then add the dilution effect. And then depending on what number you choose here, you will see some more changes. So first of all, you can just change the sample size and say, I don't want to analyze my data now. For whatever reason, you think that you shouldn't touch the trial at all. And you just want to stick to a fixed sample design, but you assume that there is a bit of dilution due to the pandemic. And then you need to add roughly the same 90 patients in order to get the power back. Or if you additionally change to a group sequential design, you would need roughly 200 patients more in order to make up for the change in the design, but also for the dilution effect. So here's my short summary of the 3 approaches discussed so far. I'd like to point out this is all based on blinded data. So as long as you have blinded data or have not looked at the data into the data at all, these changes are possible, but they are all based on basically assumptions. So what I'd like to point out here is that none of the approaches I have shown here uses any data at all. It is based on assumptions and based on maybe safeguarding your trial against some changes that you might expect, but it's not based on the data. And I'd like to point this out. I came across it while I was writing this talk, actually, that the question about the blinded or not unblinded data, I was wondering what we can learn from the data we have collected so far. Without the pandemic, we would have probably never touched our trials. What does the data tell me about the future of my trial that I've collected before the pandemic? And personally, I would think, yes, it can tell me whether my treatment is working or not, but this is independent of the pandemic because the data is not affected by the pandemic up to that point. So for me, there wouldn't really be any reason why I would look at the data when I make these decisions. It's mainly anticipating an effect and not so much about what have I seen in my data, because all I can learn from that data is how my treatment was working so far, but not really how it will work in the future based on the changes maybe in the population and so on. So for me, I would look into the data, but still to complete Chris's question about would I even use the unblinded data? So this would be the second scenario. So the investigators had taken a look at the current data, and after they've done that, they ask for your advice. So the short answer can be found in a paper by Sir Ronald Fisher. To consult the statistician after an experiment is finished is often merely to ask him to conduct a post-mortem examination. He can perhaps say what the experiment died of. It's in one of his presidential addresses. I like this quote. I quote it many times to clinicians when they talked to me a bit too late after they have already done something. So this is the short answer by Fisher. I also came up with a bit of a cartoon that might make it a bit less bitter for the clinicians when they ask you later. So here's the situation. There's a statistician, and then there's the clinician, and they basically say, okay, we've looked at the data, we have seen something that's significant, and all you need to do as a statistician now is to find a justification for the change of the trial design. And at least for a frequentist, this is how the cartoon would continue. The type 1 error is probably out of the window. It's not controlled anymore. Okay, so this is a kind of not so serious answer. Let's be a bit more serious about the topic, the statistical answer. So it's in our paper as well, so it's marked it here. So basically, as I said before, it should be noted, however, that it's necessary to specify how any data before and after the interim analysis are combined before the first interim analysis is conducted. Or in other words, if you want to change something, you have to do it before you looked at the data. If you want to change it after you have looked at the data, you run into a bit of a problem, and you will find it in the literature. It's based what's basically in the book by Gernot Wassmer and Werner Brannert. So you find the situation where they say what will happen to the type 1 error rate if you have unplanned changes, and they considered a situation where the investigators did not pre-specify the adaptation rule at all, and they later analyzed the data just in the naive way. And then they have worked out what the maximum type 1 error rate is. And then you can see that basically the first part of this maximum type 1 error rate is already alpha, and then you add something to it. So it's basically always inflated. And then for our trial example here, with an alpha of 2.5%, and you have basically your naive critical value of 1.96, you can basically work out that the maximum type 1 error rate is now 6%, or a bit over 6% even. And probably even when you allow for a bit of an inflation, 6% is maybe not really close to 2.5% at all. I have heard sometimes, or in papers, you sometimes find that maybe an inflation of up to 2.6% is still acceptable, but 6 is basically too high. It wouldn't even be acceptable when your original alpha was 5%. So what you could do now is you can say you anticipate the worst inflation you could have, and then try to correct your significance levels, and say you correct your alpha to find basically an overall type 1 error rate maximum that's controlled. And here you will find out that you basically have to use a U value of 2.35. Or if you calculate back the alpha, you have to basically switch to an alpha that's less than 1% in order to control for any changes. It should be said here that the assumption is, and I discussed this with Gernot Wassmer, the assumption is really that you allow for any adaptation that you can think of. It's not just a mere sample size recalculation or just a change in the trial design through group sequential design. It's any adaptation you can think of. And it's very conservative. So as we can see, so maybe it is possible to narrow down the type of adaptations you would allow and work out some maximum, saying that you only wanted to change the sample size, but not additionally have several looks or change the population and so on. We discussed this only briefly. We haven't really come up with the result there, but it should be kept in mind this is a very conservative. And just to show you why it might be possibly the worst option you have, if you compare the critical value that you get for these unplanned changes, so you get 2.35, but if you compare it to a Pocock or a Brian Fleming design, you see that it's much higher than the critical values you would have gotten out of these designs. So my advice would be don't do it. As a statistician, we are not always the ones who makes the decision, but if someone's asking you whether they should just look at the data and later decide what they want to do, then I would always recommend to truly think about whether there are other options available, because it ends up to be very conservative. So I think in general, there are some points to consider before you make a decision on your trial. Basically, gather as much information as you have about your current trial. So for me, that is like how much data is available, how realistic were the planning assumptions. As I said before, the power you will get will depend on the true effect and not on the assumed effect at planning stage. So how realistic was the planning assumptions when you set up the trial? Do you believe there might be a dilution effect, or do you think your trial is completely unaffected by COVID? How much missing data will you have on top of the loss in data you might have at interim? There's still missing data, and will it be more now due to the pandemic? You might have started off thinking 5 to 10%, but maybe the pandemic and all the lockdown and other rules mean that there's more missing data now. So collect as much as you can about your trial, and then explore different design options. Do not just go for I stop my trial now, or I change to group sequential design. Try out different options, discuss pros and cons with the trial team. And it could also include to basically, if you have very little data collected so far, maybe just discard the data and restart the trial at a later time point, and that might still be better than mixing data from different stages with different populations. And when you have done all these exercises, then you might come up with a good decision on the trial design. So like to end up with my final advice, changes to ongoing trials due to COVID-19 are unplanned, but external to the trial. So if you think about type 1 error, it will be protected, but you still should think about it that maybe regulators or people outside of your company, of your university that are not really into this trial might still view it very critically when you start to play around with your trial design. Then, as I said before, many design changes are possible if you pre-specify them. So that is basically makes the decision before you look at any data. You could argue that you can at least look at the blinded data, and there are different opinions out there whether or not this is acceptable. I would still think about, do you actually need to look at the data before you make decisions about your trial? Consult an expert if you're not an expert yourself, if you're unsure, and even if you are an expert, it's always quite useful to discuss with colleagues before you touch a trial, because once you have done it, there's not really a replay button or a way to go back. If it's done, it's done, and you cannot just later claim, I haven't done it, and do something else. And I also think that while I have given a frequentist view on the situation, as I have been asked by Chris, there is no right or wrong. Every trial is different, and what might be the right choice for one trial is not necessarily the right choice for your trial. So with that, I'd like to thank everyone and give it back to Adam.
Thank you, Cornelia. I don't see any questions in the chat if anyone wants to add anything, but I did have just a couple of questions. So if no one else types anything. So you mentioned at the beginning the sort of idea behind stopping just with the 300 patients in each arm, that you can appeal to MCAR data, and then you mentioned later on, when you're talking about the blinded versus unblinded data, that what happens after the pandemic has started may not be the same thing that happens pre-pandemic. So is that really then MCAR data if I sort of were to think that the data after the pandemic is different from what I would have collected in those first 300 in each arm? So specifically, what is the difference maybe it's a question of what's the difference between what your test actually tells you, because if it doesn't include the data, any data after the pandemic, it may not be the same.
Yes, it's a very good question. So I was mainly talking about type 1 error and why type 1 error is controlled, because if it's truly MCAR, then it's MCAR, and that will not affect the type 1 error. A totally different question for me would be, let's say you end up with a significant trial based on the data you have collected. Do you think you can basically apply the trial results to the population? Because what you have seen before the pandemic might not be the treatment effect you might expect later with the pandemic going on or even after pandemic. I guess that's, for me, that's a bit of a different question. So I was purely referring to type 1 error there. For me, it's still a very open question. Even if I have a significant trial and I see an effect size, do we think that this effect size is what we will see if we then go to the market and use this drug in the population? It's a bit what we always see in real world trials. It's not always what we saw in the, and this time it's really unclear. But for me, though, I only made the comment because of the type 1 error.
Okay, so now there are some questions. So hope I don't say this too wrong. Jing Rong, she's asking, or he or she is asking, if before any data is analyzed, does that mean even SD is not available? So can you use the standard deviation from your 300 to help you calculate or maybe adjust the sample size? Did you even avoid the standard deviation, or do you feel that that might be more reasonable?
No, as I said before, I mean, you can look at the data, but for me, the question was mainly, what do I learn from data collected before the pandemic about the data that I collect during the pandemic or after the pandemic? Because if I believe it's exactly the same, then why would I look at my trial? Without the pandemic, I wouldn't have done this, right? Without the pandemic, I would have never thought, I need to look into my data now and I need to make changes here. So if the pandemic would have never happened. And now there are two options. I say the pandemic has happened and I can't see a reason why it would affect my trials, and why would I look at the data? And if I think the pandemic is affecting my trials, then what can I learn from the data I've collected before the pandemic? So for me, it was kind of like, it's either or. If I believe there is an effect, then I can't really use the data I've collected so far, and I have to make assumptions. Or I believe there is no effect, but then why would I touch the trial? Then I wouldn't have done it otherwise either. Of course, I can use the opportunity and say, because there is a pandemic, I'd like to look in my data, and maybe something was conceptually wrong with my trial, even without the pandemic. That might all be the case, but it will add further to what I said on my last slide. Every time you touch a trial, it's viewed critically. People will ask, why did you touch the trial if you didn't believe that there was an effect? And I think it's kind of like, it's a difficult situation to be in, to say, how much do you change of the trial now, and how would you explain that later if people question that? But it's possible, of course. It's possible to look at the data and do a blinded sample size re-estimation, but you have to basically be aware that whatever you observe in the data will not tell you something about the future data. It tells you something about the past data.
Okay, thank you. And there are other questions, but perhaps we'll leave them because we need to move to the next speaker. So we'll perhaps address these additional questions in the discussion session after this session has ended. So this brings us to the next talk. Will be by Gary Rosner at Johns Hopkins. And he's going to sort of, I think, talk more from a Bayesian perspective. Cornelia did the frequentist perspective, so now we will look at more of a Bayesian perspective about how to answer these questions. Thank you. Go ahead, Gary.
Okay. Thank you very much, Adam, for the introduction, and thank you to the organizers and to Chris. Let me just say that Chris Jennison and I were working on group-sequential designs as part of our dissertations quite a while ago, and it drove me more to the Bayesian side. I'm not sure that it had the same effect on Chris, but the gymnastics one has to go through on frequentist study designs, and the concern about type 1 error just seemed a little, let's say, unscientific to me. Anyway, I'll try not to be too preachy, but I'm going to talk a little bit about Bayesian statistics and clinical trial disruption. And for the most part, I'm considering the scenario that Cornelia already discussed. And so this would be a design that was a trial that was designed with frequentist characteristics in mind and a null hypothesis significance test at the end. And so we're concerned to a large extent about the effects on the frequentist characteristics and how a regulator might view the study. So how what might a Bayesian approach help with respect to a study that's been disrupted for reasons of like a pandemic? So some active nature. So one of the obvious things that one can do is to carry out an interim futility analysis, regardless of the type of the design. And as I'll show, that has virtually no effect on the type 1 error. But one would use the predictive probability calculation. So I follow the approach that was presented a long time ago in a paper by David Spiegelhalter, Larry Friedman, and Blackburn, I think, was the third author, arguing in favor of predictive power as opposed to conditional power. And the reason is that we have information already in hand, and why not use that information? And as I said, if we're doing a futility analysis, it's not going to affect the type 1 error probability. And now the question, of course, is with respect to what prior, and I'll talk a little bit about that, but I think Chris may have something to say about that as well. Another way that one might want to consider applying Bayesian approach would be to augment the study data with external data. And we heard some of the earlier talks in the forum have talked about this. So Dr. Lily Yui of the FDA talked about using propensity scores, as well as a more recent talk, I think at the last session, about using a power prior approach to bring in external information to bolster the sample size and try to overcome the loss in power that happens because the sample size has been cut short. And then Mark Vandenbroucke also talked about the approach at Novartis. They talked a little bit about this. Another thing that I'm going to talk a little bit about is change in patient population after the disruption. So there would be a drift in the patient characteristics over time. And doing sensitivity analyses. And again, where a Bayesian approach might help is that a treatment effect, it seems extremely artificial, and I don't think anybody truly believes that the treatment effect is going to be exactly 0. It's going to be exactly 0.2 in our example. And so there should be some sort of distribution about the value, to characterize the uncertainty. And so the Bayesian approach can allow one to consider sensitivity to deviations such as these. So what are some of the questions in light of the disruption? Well, what might happen going forward? And Dr. Kuntz was saying that what happened pre-pandemic, say, doesn't tell us anything about what might happen in the future, but I would disagree. And the Bayesian approach one of the strengths of the Bayesian approach is that there's a clearly defined predictive distribution that obeys the rules of probability theory. And it basically talks about the distribution of future observations given the current ones. And the clean definition would just integrate the likelihood or the sampling distribution for the new observations with respect to the posterior distribution given the current. But one can also perturb the prior that one uses for these new observations, as I'll talk about later, and still make predictions, and the predictions are still going to be valid with respect to probability theory. And that would be very useful for sensitivity analyses, as well as planning purposes, because one can also use, or one would use, the predictive distribution if one were going to use some sort of decision analytic approach, taking into account risks and costs of continuing the study and so on. So this is a very powerful tool, I believe, that the Bayesian approach provides, and that should not be dismissed lightly. So again, conditioning on the actual data brings us to the Bayesian predictive distribution. But if we condition on a hypothesized parameter, such as the dilution effect in future observations, but assuming a fixed dilution effect, although I did notice that the Shiny app also allowed one to make some changes to the variance term after the disruption. But conditioning on the hypothesized parameter is frequentist. So we have information at hand, and how one uses it is clearly a question. So one question obviously is, should we continue the study? Now, what is the chance that the study will show a difference? And even if the study design is based on Neyman Pearson hypothesis testing, one can still use Bayesian inference at the interim, in particular for futility analysis. Is there any point in continuing? Because it seems unlikely that we will see have high probability of seeing a significant outcome at the end, significance being the hypothesis test. And there are several examples from Bayesian inference at the interim, but I'm just going to talk about the continuous outcome. But I have used it in the past for a binary outcome and also for time to event, using Bayesian non-parametric approach in the latter. But the example we have to work with, we have continuous outcomes, namely the change in this marker of glucose level among diabetics. So the example randomized clinical trial, just to remind you, although Dr. Kuntz already did talk about it, we have a randomized trial. Patients are randomized evenly between metformin and sulfonylurea as the control, the standard treatment, against metformin and a new drug added to it. And the endpoint is the change after 6 months in the HbA1c level. The type 1 error probability is set at one-sided 0.025. The power that was specified in the protocol was 0.9 for an effect size of 0.2, assuming a variance of 0.95, and we have a sample size of 500 patients per arm. So that's what's in the protocol. So recruitment started October 2019. The trial was disrupted 13 months ago, March 2020, and they reached 300 patients per arm. In March, accrual stopped, but they continued follow-up. So we have 6-month evaluations for all of the 300 patients who entered prior to the disruption or the stopping of accrual. So should we analyze the current data? Again, these are the questions that Chris Jennison posed to us. Should we analyze the current data to determine efficacy of adding the new drug? So there's the randomized comparison. Should we restart recruitment, perhaps change the target final sample size? It's not clear when the studies will begin recruitment, and time taken to reach the final decision is an important consideration. And this is a place where I think decision analysis, that is a utility-based approach, would be beneficial in terms of the trial delay, what the cost would be to restart things. And that's, again, where a Bayesian analysis would clearly be beneficial. But I'm not going to be going into the details of that in my 30 minutes or 25 minutes with some questions. And if the data are currently blinded, we could develop procedures to allow stopping now or continuing, depending on results when the current data are unblinded. And so, you know, there are some ways of dealing with the blinding issues, such as having independent individuals, if one specifies a particular analysis, like a futility analysis, what to do. But anyway, blinding versus unblinding is always a question. So choices for interim analyses. As I mentioned before, I would use a Bayesian predictive approach. Using the predictive distribution, that is sampling distribution for the future observations, conditioning on what we currently have in hand. And one approach that I think is very helpful in terms of setting the priors would be going back again to Spiegelhalter and Friedman's approach of having both an enthusiastic and a skeptical prior distribution. So the enthusiast is someone who a priori believes that this new drug added to metformin is going to improve outcomes for these diabetic patients, where outcome is with respect to the study's endpoint of change over 6 months in the HbA1c level. And so we would want to stop for, if the current data, if the prediction that the future study, the future data will lead to the enthusiast losing almost all enthusiasm, so that the enthusiast is no longer as enthused about the prospect that the new treatment adds benefit. But we might continue if the skeptic gains considerable enthusiasm. Likewise, we might want to stop for futility if the skeptic has now, we might stop for futility if the enthusiast has lost their enthusiasm. We might want to continue if the skeptic actually gains some enthusiasm, so that it's quite possible that by the end of the study, the skeptic would be close to the enthusiast's belief. And one approach that I'll be talking about would be using Zellner's G prior, which is a so-called objective Bayesian approach. And this would be in the situation where we have we want to carry out the analysis of covariance, which in this particular example, the way Professor Jennison set up the data, the baseline values of the biomarker, of the measure of glucose levels in the blood might not be that beneficial, but in general, there are plenty of papers that show that adjusting for baseline characteristics often does improve power. So I'll talk about using Zellner's G prior. So stop if low predictive probability of ultimately rejecting the null hypothesis, or continue if there's high predictive probability of ultimately rejecting, where what is considered low or high would be something that would be pre-specified what this threshold might be. So how would we specify the skeptical and enthusiastic priors? So one approach might be to use the information that was already set in the protocol with respect to the frequentist characteristics. So we could calibrate our prior normal distribution for delta, the change over 6 months, via this alternative of an effect of 0.2. So the skeptic's prior distribution for delta, this change over, this difference between the two treatments and the change, 6-month change, would be centered at 0. That is, there's no treatment difference, but you'd set the skeptic's variance to some value that would say that there is maybe only 2.5% probability that it's greater than this alternative of 0.2. And so you can set, just using the frequentist, the assumptions that were made in the trial's design, to set the prior distribution for this, the prior variance, and we know the mean. So the prior normal distribution for the treatment difference for the skeptic. And for the enthusiast, we might center their normal prior on the alternative of 0.2. And again, use the information in the protocol to specify the variance for this normal prior for the enthusiast. Again, based on the power considerations, or the type 2 error probability. So what I did was I simulated some data where I was simulating the full 1,000 patients. And I simulated them with respect to a particular setup that Chris Jennison was kind enough to give me for both a baseline value as well as a 6-month value. And I have to admit that I did several simulations until I got one where the interim analysis did not reject the null hypothesis, but the final analysis did. So this is a simulation for the particular example we have, but it's to some extent contrived. So we've reached 300 patients per arm out of a planned 500 per arm, so 60%. The 6-month endpoint is recorded for all 300 patients. And for the particular example, let's assume that for the metformin and the standard arm, we have a mean of 1.5 for the change over 6 months, a certain standard error, 0.06, and there are 300 patients in each arm. And we can see there's a slightly greater change for the new drug combination with metformin. And so now at this interim analysis, delta hat was 0.109, so 0.11, not quite 0.2, and the standard error was 0.0776. And if one did a t-test at this point, one would find a one-sided p-value of 0.081. So that just gives you an idea of what a frequentist would do if the frequentist analyzed the data now. So I've also carried out posterior inference at the given data set. And so here in this graph, I'm showing what the skeptics and enthusiastic prior distributions are. Those are in these dashed lines. And I just want to see if I can get my, yeah, I guess it's a different Zoom setup because I can't show my my little cursor thing to point out different things. But you can hopefully see that there are the black dashed line to the left corresponds to the skeptics prior, and that's centered at 0. And the one to the right is the enthusiasts prior, which is centered at the alternative 0.2. And at this point, we can see that the solid lines correspond to the skeptics and enthusiasts. This is the predictive power if the study were to continue to the end. I'm sorry, these are the posteriors. And so they're starting to come together somewhere in between the two prior values, but they haven't really joined that much. But if we make prediction, assuming that the data after disruption is the same as the data before, that is, we're predicting the next 200 patients per arm, conditional on what we've learned from the current, we get the predictive power for the skeptic of 0.3 and for the enthusiast of 0.53. So depending on what threshold one has set, one may or may not want to stop the study. So I just arbitrarily said that if the enthusiast's predictive power is below 1/3, then we might want to stop the study. So it's not, it's around 1/2. So probably we would continue and not stop for futility. So that's just doing a simple analysis based on the treatment difference. Okay, so that's not predicting future values, but it's predicting the future treatment difference. What if we wanted to actually do an analysis of covariance? And so we want to predict future observations. So a common objective prior is what's called a unit information prior, and that's basically looking at the information that would be contained in one unit of observation and using that as our information in our prior distribution. So if our prior uses the covariate information, as we would in the variance of the regression coefficient beta, then ordinary least squares we know is given by the formula here. And we know from our regression classes that the ordinary least squares estimate has a normal distribution centered at the true regression coefficient vector and variance of sigma squared times x prime x inverse. So with n observations, we can compute what the information is. It's just the inverse of that variance. And so the unit information just divides that by n. So Zellner proposed what he called the G prior, which is instead of dividing by n, we just use some particular constant G. And in fact, what I will be using would be the sample size n, so unit information. But if we use that prior, we center the prior mean for the regression coefficients at 0, at least for the regression coefficient for the treatment effect at 0. Then things work out very nicely with this G prior, namely that the posterior mean is just G over G plus 1 times the least squares estimate, and the posterior variance is G over G plus 1 times the variance corresponding to the least squares estimate. And so conditional on the data for the first part of the study, that is the 600 patients, 300 per arm, we now can compute a regression estimate, least squares estimate, and use the G prior to then make predictions for the next 400 patients if we continue this way. And it's very easy to show what the posterior and distributions are and how we can plug in the values from the first 600 patients. And if we do that, we can then use a simulation-based approach to generate the predictive values, or use a simulation approach to look at what the predictive distribution is if we continue the study, again, conditional on the first 300 per arm, combine that with the initial 300 and compute the ultimate test of the regression coefficient corresponding to treatment, conditional on the baseline value. And so the model that I use that Chris kindly provided gives me a normal distribution for the baseline HbA1c values, as well as a value for the 6-month measure in these individuals, and which is here where there's a treatment effect of 0.2, assuming the alternative that was in the protocol. And it has these characteristics in terms of that are shown at the bottom in terms of the mean treatment difference, the variance of that, the covariance of the treatment difference with respect to the baseline value, which we see is relatively small. The correlation is about 0.2. And so if we do simulations under the null hypothesis, and again, using a 0.025 one-sided test, we see that we preserve the type 1 error probability because the simulation showed that it's only a 2.6% risk of falsely rejecting. Okay. Under the alternative that the treatment is 0.2, but we stop if the predictive probability is less than 1/3. And these simulations were done not for the particular setting we did before, but just let's say that we stop halfway between. Then we see that we would stop early for futility about 12% of the time, and we would reject ultimately 86% of the time. Now that's lower than 90%, but I just note that even without the interim analysis, it's still about 86% of the time we stop. So in these simulations, the actual achieved power would be closer to 86% than 90%. So doing this interim analysis preserved the type 1 error probability and still allowed one to make a decision about early stopping, but didn't really alter the characteristics, the operating characteristics from a frequentist standpoint. So for the particular study, if we did carry out the analysis using a G prior at the interim, what we would find is that the beta hat for the treatment, that is using this analysis of covariance, is 0.099. And so the p-value one-sided was a 0.099. That's if one did the actual analysis. But if we use the predictive power, it's 0.38. And so using a criterion such as stopping, if it's at or below 1/3, we would continue. And so at the final analysis, in fact, we see that with this particular data set, because remember, I simulated a specific data set of 1,000 patients and just took the first 600, But if we had, in fact, continued, the ultimate p-value for the treatment effect would be 0.019. So it would have reached significance. Now, the other question that arises is, what about the fact that patients after the disruption are going to be different? So one could adjust via covariate using time interval, or one could use an approach that's similar or that borrows from this idea of a robust meta-analysis predictive prior, where you take the information conditional on the data, but we add 1 minus epsilon. So epsilon times that, where epsilon is just some number pre-specified between 0 and 1. So we have the posterior distribution for the data, some proportion from that, but then we mix in a little bit of noise. So 1 minus epsilon times p0 theta, which could be a robust or a much more vague prior. And then the prior, after the disruption, that is the prior we would apply for making predictions for patients after the disruption, or even at the end of the study to do a sensitivity analysis, would just be a mixture of the posterior pre-disruption and some extra noise to allow for lack of exchangeability of the patients before and after. And so just schematically, it could look something like this, especially if we wanted to break up the time of the trial into different intervals, where at each of these particularly pre-specified, but clearly time intervals where there could be lack of exchangeability, we could just continue to add some noise and apply to the posterior at the start of an interval when we analyze the interval specific data. So we would add or contaminate to some extent the information coming from the previous interval, contaminate that with times 1 minus epsilon 1, some small value, but pre-specified of the vague prior. And so at the end of the analysis, we control for drift in the patient population and allows for the patients no longer being fully exchangeable. So in summary, a Bayesian analysis at the disruption, I think, is extremely useful for decision making. And again, I'm assuming that the study wasn't initially designed as a Bayesian study. Had it been designed as a Bayesian study, a lot of these same techniques might apply, but there also might have been other things that were built in that would allow one to be a little bit more robust. We can see that there's no inflation of type 1 error probability with futility analyses, and we can use the Bayesian methods for sensitivity analyses to allow for drift or changes in patient populations or patient characteristics, pre-pandemic, say, and post-pandemic. And that is it. Thank you very much.
Thanks, Gary. I had one question about G. So you use G equals 1, which I think makes sense to me because it's essentially 1 unit information for each patient. Or
G equals N.
Yeah, or G equals N. Sorry, what equals N is in in 300 or 500? Is that?
At that point, it was 300.
Okay, so it's 300, and then it would go to 500.
Well, 300 per arm, 600. Yeah.
Right. Yeah, and so then. What would be an alternative G? I mean, so would it always be 1, or is there any reason for alternative suggestions of G?
Well, the larger G is, the greater the strength in the prior distribution. So with 600 patients, whether it's 1 or 20, it's not going to make a big difference. But in general, we like to make G small.
Okay.
Well, I shouldn't say small, but so that it has the effect of a small prior sample size. Right. Let's put it that way.
And so Nigel, this is probably the last question. Nigel asks, so performing a futility analysis of the 300 per arm seems sensible, but if you analyze these data, it is hard to believe you wouldn't claim a treatment effect without restarting if you observed a very large treatment effect. Would you consider combining the Bayesian futility analysis with a frequentist group-sequential design so as to allow a stop for efficacy without inflating the type 1 error rate?
I'm going to assume that Nigel has in mind changing the design at the interim analysis to become group sequential, and then specifying some kind of boundary from then on that's based on the Bayesian approach. I mean, because one could do that with respect to the skeptical prior. One could set up the boundaries so that it has the appropriate kind of stopping, and through simulation you can calibrate things so that the operating characteristics are what one wants to have, say, for regulators. That's something that Peter Thal and colleagues do all the time, is to calibrate the distributions to ensure good frequentist characteristics. I'm thinking that that might be the sort of thing you have in mind, but please clarify if I've gotten it wrong.
Yeah, that was sort of what I had in mind. I suppose, I think what you're proposing is a futility analysis based on the Bayesian analysis. And I suppose I'm sort of saying it's hard to me to see that it's convincing that you look at the data and you say there's no chance of stopping early here because of a big effect. You know, supposing when you looked at those data, you know, the Frequentist p-value was, you know, 10 to the minus 11 or something. You know, it's so tiny.
Right.
You know, are you really saying that in that case you would simply say that my futility analysis hasn't stopped the trial, so I'll just restart to collect some more data? And if you're not saying that, then where's the cutoff between stopping or not? And don't you have to specify that to control the error rate?
Right. Well, one could certainly have done that. So before you do these enthusiastic and skeptical predictive power calculations, you can specify what the cutoff is. But as you know, one would want to be extremely conservative because one doesn't want to end up declaring a treatment effect and having no one believe it. You know, I'm always cognizant of ECMO and the number of times that particular treatment was reevaluated because people didn't believe the results.
But doesn't that take you back exactly to the sort of analysis that Cornelia was talking about?
It does to some extent. And I think the Shiny tool and so on would be very helpful from that. The advantage of this is that, you know, you can calibrate or by pre-specifying the cutoff. Hopefully it would be convincing. And it would seem to me that a 600, of course, I work in cancer where most of our studies are not that large. And so a 600 patient study is a fairly large study, but I guess relative to the effect size, it wasn't that large. So yeah, I don't know. I don't know what people would believe. It'd be worth, when trying to decide, to calibrate these things with other experts in the disease, as opposed to just letting some ignorant statistician make the decision.
So I think we needed to have some time for a quick break. Perhaps Jim will tell us the details of what to do.
Yes, we will. We'll give everybody 5 minutes or more to take a break. We'll try to stick on to the schedule, and that brings us back at 25 minutes after the hour. And there's the schedule. So I suggest you go take a break. If you want to chat with each other, turn on your camera, and those in the room can chat. I will stop the recording during the break. So thank you. Thanks for these great talks, and I look forward to, Chris, your discussion. Following that, we'll have the breakout rooms.
Shall I share my slides?
You can share your opening slide. And Adam, are you going to say a few words?
Yes. So the last speaker will be Chris Jennison from the University of Bath. And if, like I said before, he proposed this question that both Gary and Cornelia have been investigating, and this will be his talk about his thoughts on their presentations, his discussion of their presentations and the problem in general. Thank you. Chris?
Thank you, Adam, and thank you to the speakers for really interesting presentations. I've had a chance to see their talks and try and write some discussion. And here's an outline of what I'd like to discuss. So we've got our example, and in a little schematic here, we've got our 600 patients recruited and, well, responses maybe not observed at the time of disruption, but follow-up will have brought them in later. And then the questions, do we stop? Do we restart? If we restart, do we redesign? And I think most of the attention by our two speakers has been on the first case of the investigators being blinded to the results so far. And as Cornelia said, it's really desirable that that's the case. But we can also consider the other situation and think about how you might deal with that. So in the first case, what are the options? One can stop and either make a decision, make a declaration that the new treatment is found to be superior, or stop for futility and not make a claim that the new treatment is superior to control. And then we can continue. And if we do that, perhaps modify the final sample size. I think that can mean different things. We can modify the final sample size relative to the initial design. We could modify it based on the current treatment estimate. We could modify it based on an estimate of the variance that came up in discussion. So I'm going to look at this from two different approaches. First, how do you do this if you're being purely frequentist? And then how do you do this if you're being out and out Bayes? And let's compare where we get to. And the ingredients, to start with, they're the same ingredients in both cases. We have our observations. So with mean mu t on the new treatment, with mean mu c on the control, and our treatment effect theta is this difference. And the null hypothesis is that theta is negative, so the new treatment is not superior to control because we're trying to get a good reduction in HbA1c. And the alternative is that theta is positive. So now into the frequentist domain. If we have set type 1 error of 0.025 and a power requirement of 90% power if the treatment effect is 0.2, then we've got our usual formula for the fixed sample size per treatment that we would need. So as Cornelia said, we can simply look at this situation at the point of disruption. And since we haven't looked at the data to see any of the response information, we can imagine we're actually just designing the study as if at the beginning, even though we've reached in calendar time, we've reached this disruption point. So how do we set the stopping boundary? And if, when we continue, there could be more analyses. We don't have to have a single interim analysis at the moment. We could have later interim analyses if we're going to put them in. And we could have a new final sample size. Gary has focused on a futility analysis at this point. And we can think of that in the context of the frequentist approach. So the principle here is to stop early if the predictive power is low. And this won't inflate the type 1 error rate because the only change to the original plan will be to halt the trial with a negative result. So there can't be any excess false positives. The question in this case then is, how do you choose the prior under which this predictive power is calculated? And what threshold do you set for this low predictive power that's going to lead to stopping for futility. I should say this is a question that comes up in, if you like, standard group sequential design, particularly when people work with what's called a non-binding futility boundary. So you may set your efficacy boundary in some fixed way and then have a more informal approach to stopping for futility. And people may say, well, we'll look at the predictive power or even the conditional power at some effect size, maybe the original delta at which power was set, or maybe the current theta hat. And I think the problem then is there are so many different ways to think of conditional or predictive power, and there are so many possible thresholds to set for it that you've still got, a serious choice to make. So how do you go about making these choices, particularly, first of all, in the frequentist domain? In the example, a lot of attention is really focused on a group sequential design with two analyses, because there's the disruption point and then there's the period after that might be taken as a single stage of the trial. I think it's more informative to think about what's going on in a sequential design when there are maybe 3 or more analyses. So let's take a more general case here with capital K analyses in our study. And then we'll be looking at our test statistic ZK, which will be mean 0 under the null hypothesis at each analysis. And we can choose a stopping boundary to satisfy the constraints, the type 1 error alpha, the power 1 minus beta if theta equals delta. Right. So there's standard theory and there are standard methods of doing the numerical calculations to achieve this. And of course, there are a variety of boundaries. We've got 2 capital K minus 1 boundary points here, but only 2 constraints. So there are many boundaries that will achieve these error rates. And then the question is, how does one go about choosing between them? So we can add in another consideration and say we'd like to work within a class of boundaries and minimize sample size in some sense. So expected sample size, and we have to say where. So in this first criterion, I've taken the expected sample size at a treatment effect of 0 and then again at a treatment effect of delta and taken their average. I could integrate expected sample size against a weight function, W of theta, which reflects both the values of theta that I think are likely to happen and also how important I think it is to stop early under particular values of theta. If we go back a slide, I've set up my final sample size, my final analysis with a sample size which is greater than the fixed sample size by a factor R. If we are going to have any early stopping and still achieve the same error rates, R has to be bigger than 1. And then I can space my analyses at times in between. I'm not so worried about the spacing here. Equally spaced is a simple common assumption. So if I choose R and I choose my analysis times, then I can look to find the boundary which minimizes one of these criteria. I'll say a bit more about how one does the calculation of that later on. And I like to do things in that order, because once we've done that, we can then see, well, what would happen with different values of R and different numbers of analyses K. And what you will find is that as you increase R, you can do a bit better in reducing expected sample size. And as you increase K, you'll reduce expected sample size. And so you have to decide on the trade-off between a higher maximum sample size, possibly more analyses, and the reduction in E of N. One can go further. This really hasn't come up in discussion. If you want to make your group sizes depend on the data seen so far. So this is the theta hat at the moment. One can also do that, and one can optimize in that larger set of designs. And one of the reasons that Bruce Turnbull and I did that was to demonstrate that actually it was rather pointless and you didn't gain very much. So one can actually simply stay with group sequential designs with fixed groups, and you will not be missing out on anything very much. So those are the ingredients, I would say, of the frequentist approach. And we've got our alpha and beta. The setup of the design is really what it has to be. The one thing we have to specify in arriving at a boundary is the function of expected sample size that we're going to minimize. So what do you do if you start work on the problem from a Bayesian perspective? So we've got the same model for our data. What would one do? Well, you should have a prior. And then here I'm following a Bayes decision theory approach. So I'm throwing in really everything in the kitchen sink to get a complete problem. And I've made a function, a loss function, to minimize. So the first term here is to do with making a type 1 error. So if theta is actually negative or equal to 0, and I reject h0, I have a penalty, lambda 1 of theta. Then the next term is to do with a type 2 error. So if theta is actually positive, and I should be rejecting h0, but I don't, then I'll have a penalty for that. And finally, I've got a term to do with the cost of sampling. And in all these cases, I integrate over the prior, because that determines the appropriate distribution for theta, and then I multiply by a relevant cost. Gary mentioned that in setting power, you should really think about a distribution for theta and think about power under different theta values. So that's being taken care of here in this second term. And I say I like this approach. I think I'm a bit ambivalent at times as to whether I'm frequentist or Bayesian. But if I'm Bayesian, I want to do things like this. I'm not so keen on a stopping rule that says we look at our posterior distribution for theta and decide if it's extreme enough, because I don't know how to decide what extreme is. Whereas this framework, really, it will involve at any stage, it will involve the posterior distribution of theta, but it's got criteria against which to set that. Basically, is it worth the cost of taking more information to reduce the probability of making an error in my decision? So what goes into this formula? As I said, there's a prior, and I would assume that to be the investigator's prior beliefs. So it's a really subjective prior. And then, well, actually the tricky part is how do you set your lambda 1, your lambda 2, and your C? They're to do with quite different costs of treating patients and the costs that come from making an incorrect decision. So to put those on a common scale is something of a challenge. One can think of it maybe more as a matter of how do you decide on the right rate of exchange between having more patients in the study and making more accurate decisions at the end. Of course, it turns out, I would say, this is a very difficult problem to address, and in the end, we don't really need to address it. If you do try and put in costs and so on and get a procedure, someone's going to ask you, what is the type 1 error rate? What is the power at a certain effect size? And if there's a phase 3 trial, the regulators are going to insist that you control type 1 error at some specified level, typically 2.5% one-sided. And the sponsor, the trial sponsor, will no doubt have a view about obtaining adequate power at the anticipated clinically significant effect size. So you may find you're subject to constraints. In fact, I'm sure you will. In which case you have to go back and tweak your prior and your cost functions so that these constraints become satisfied. In the case of a two-stage trial, there are only 3 degrees of freedom. Once we've set the inflation factor R and say decided to have the first analysis halfway through the study, there are only 3 boundary points, and we've got 2 constraints. So there's 1 degree of freedom. And I think every boundary is the solution of some problem or the solution of a great many problems, which is why I say it's rather more interesting when we've got 3 or more analyses. I have a comment on the role of the prior here. We know in a fixed sample analysis, say, the prior can have a significant effect on the inferences drawn from the data, particularly when its weight is on a par with that of the data being collected. And I would say in clinical trials, when you're trying to stop as soon as you have the right amount of evidence to make a decision, then that can be a situation where the prior could play an important role. But once we calibrate, once we say that we're fixing our type 1 error and type 2 error, then we have probably built something into the prior to do that. And there's not much freedom left to modify the prior around some sort of skeleton version to have much of an additional effect on inference. And I think where the prior really is therefore coming in is in terms of the properties of the design that we're trying to optimize. So if we go back a couple of slides, we see this third term here, which is our, if you like, cost of sampling, pulling out which values of theta do we most worry about, either in the prior or in the associated cost. And they will affect the design. And I think the same kind of phenomenon is appearing in analyses or derivations of adaptive designs in other contexts these days, where you can set up certain features of your design to deal with the error rates. So in the paper I reference here to do with adaptive enrichment designs, there's a construction to do to protect the family-wise error rate with adaptation using close testing procedure and combination tests. But that leaves freedom in deciding on the enrichment rule. When do you decide at the interim analysis to enrich to a subpopulation? And then that you can deal with by saying, well, I'm going to set up a utility function, have a prior distribution for the parameters I don't know, and optimize it over it. So you're seeing there, I'd say, a coming together of Bayes and frequentist ideas. So the point I would make here is that Bayes and frequentist can take us to the same place. In fact, there's a stronger statement. The 2 approaches I have described should actually lead to precisely the same set of possible optimal designs, depending on what you feed in as your function of sample size that you want to minimize. So in the frequentist case, I had this thing with a W in it. In the Bayes, I had pi times theta. So one reason for this is what is known as a complete class theorem, part of statistical decision theory, which says in almost all problems, the set of admissible frequentist designs equals the set of Bayes designs. So this shouldn't be a surprise, although I think it is to some people. The other thing that I've noticed or I've come across is in computing the optimal frequentist designs, there's a tricky optimization problem because we've got error rate constraints. So we're choosing a boundary subject to 2 constraints, and that's not a nice problem. And so what one can do is introduce a Lagrangian method, or Lagrangian multipliers. And if you look and see what you've done, you've basically, if you interpret certain things as priors, you've produced a Bayes problem instead of a frequentist one. And then you have to tweak the parameters in your Bayes problem in order that a solution satisfies the type 1 error and type 2 error constraints. So we're doing exactly what the Bayes problem is doing when it calibrates. And of course, one thing, one can look a bit more closely at those calculations and see at some interim analysis, the way in which the boundary is being constructed is all to do with the posterior distribution of the parameter theta, the treatment effect, and the impact of that on what could happen in the future. So the predictive power. So I would say there really is no conflict between Bayes and frequentist approaches. However, I think if you look, you'll find there are basically two disjoint sets of papers in the literature that work towards these designs. And of course, when they do it right, they get the same result. I won't say about what happens when they do it in other ways. So here are my conclusions on, if you like, in general on Bayes and frequentist approaches. One, I would say one can use either approach, and there are nice ways to explain what's going on, depending on your point of view, whether it's low probability of futility stopping or low predictive probability, whatever. But in order to have designs which are internally consistent, then there have got to be particular ways in which boundaries are set or thresholds for stopping for low predictive power or stopping for a positive effect are achieved. While I'm just giving my general thoughts here, I will say I do like the approach of fixing this inflation factor, which is basically determining the maximum sample size, and also fixing the number of analyses and doing the optimization, rather than trying to optimize over these things as well. That's actually a much more tricky problem anyway. But you can then look and see which values of R and K you find acceptable. And what you will, I think, normally find is that the inflation factor should take you up to maybe 5% or possibly 10% more than the fixed sample size. You need something to get your group sequential design going, but you don't need much more. And the number of analyses, I mean, a lot of people seem to stick at 2, and actually I think there is benefit still to go up to 3, 4, or 5, but certainly not much beyond that. Now, in practice, does one use an optimized design? Probably not. There are other things one likes in a design, but these can serve as benchmarks. So one can use them, for example, to help you choose a family of error spending designs that are efficient. And my final comment, I like to see things coming out of a formal, like, decision theory type of framework, simply defining in terms of futility because a certain probability is less than 0.1 or less than 0.3. I think there are too many arbitrary choices there. One should look at things in the bigger picture. Okay. Let me move on and say a bit about special features of the example that we're looking at that maybe change the way in which you would normally think about a group sequential design. And one, of course, is that there's a delay. So the disruptions happened. If the trial is going to start again, it will be at some future point. And rather than look at sample size in a stopping criterion or utility or a gain function for early stopping, one might actually look at time, because time is crucial to determining when patients benefit from a new treatment or how long a company has to benefit from its patent. And since we know there's a delay in restarting, that's actually saying there's a bigger return on an early decision if we can stop and make the right decision. So that probably tells us we're going to spend more type 1 error at the disruption analysis point than we would otherwise. As Cornelia showed in her 2 versions of her example, if we do that, then there will be implications for a larger maximum sample size if we're preserving both type 1 error and power, which come with that. There may be changes in the post-disruption phase. The speakers both addressed the possibility that either patients change or the way in which the treatment works on the patients will change. And we could expect this in this diabetes study due, for example, to lockdown behavior having a different lifestyle impact. And I think there are questions to bring out of this. I think these are open questions. I don't have any answers. But one should ask, is it still reasonable to test a single hypothesis about the treatment effect after disruption? Or have we really got two quite distinct situations? If that's not reasonable, then maybe we should simply stop and analyze the pre-disruption data, because that's addressing a well-defined question. One might then say, well, if we're doing that and we're going to think of the post-disruption data as essentially a new trial, do we get another alpha to spend on that one? So let me briefly comment on the non-blinded case. What if investigators have looked at the data and we want to help them do something while still protecting type 1 error? So that really is a challenge. Cornelia mentioned this result in the Wasmer and Bernath book, where you can try and imagine the worst case scenario and then protect against that. Now, if that means that to do your 0.025 significance test, you end up nominally testing at 0.0093, that sounds like things are going to be quite conservative. And my quick calculation tells me that in order to have the same power, but working with that smaller alpha, you would actually need a 25% increase in sample size to compensate. I think this worst case scenario might be quite bizarre, and so one would hope that one can get away with looking at less extreme cases. Gary mentioned the option of futility analysis, which doesn't inflate type 1 error, but that is on the assumption that everything continues as originally planned. If there are some possible design changes, and they can be quite subtle and not very transparent, I think we're familiar with that now in the world of adaptive designs, one would have to worry that the investigators had done something that actually increased the type 1 error rate. One way around that is to try and work with the conditional error principle as a way of allowing one to look at the data and then do something which still protects overall type 1 error. So the system here is that you ask, well, given where we are now, if the trial continued as it had been originally intended, what's the conditional probability of a type 1 error if the treatment effect is 0? Then one is free to redesign the remainder of the trial, and one simply carries out a test at the end using just the new data at level alpha prime. And it can be shown as a very simple argument that this will protect the type 1 error overall at level alpha. One thing I would say, though, is that the devil is in the detail in saying that if the trial had proceeded as originally intended, here are some questions. What if at the moment recruitment is on the low side? Do we assume we're going to have lower than anticipated recruitment at the end of the trial? Or do we assume that plans would have been made to extend recruitment or add more centers and make up the shortfall? And those two different options will have an impact on that conditional type 1 error. There may be nuisance parameters. How do you, therefore, say what would happen in this final analysis if you've got an unknown variance in the calculation? And regulators, being very cautious and suspicious, might think that the investigators have worked their way through various ways of describing how the trial would have proceeded and found the one that gave them the best, from their point of view, the highest value of alpha prime. And I think this comes under the category of Gary's jumping through hoops when you're a frequentist trying to control type 1 error rate. So I would close with the question, can the Bayesian approach help us out of this particular problem and avoid some of these issues? Thank you.
Thank you, Chris. It is right now 11:55, so I think maybe it's best if we move. We can have all kinds of discussion in the breakout rooms. So I'll turn it over to Jim to sort us into the correct groups.